Good evening, everyone. Emory Moynagh here for In His Image, and welcome back to Beyond the Basics, a creation podcast. Today we're going to be reading a secular paper that was published in Nature this year. It's a brand new paper, only a couple of days old. And the title of the paper is Mutation Bias Reflects Natural Selection in Arabidopsis thaliana. I should have pronounced that right. I've read it enough times. But Arabidopsis thaliana, it's a model organism. It's a commonly used one, used in polyploid research a lot. So we may be getting into that. We may be getting into polyploid research too. There's some nerdy stuff I want to talk about related to that, but that's a whole other topic. So this paper came out only very recently. It's got a laundry list of authors on it, and it's not short. I'm holding it right here, and as you can see, it's kind of long. But we're going to talk about this paper because this paper is very significant, and I think you all can learn quite a bit from this paper, and that's why we're going to talk about it, because I think this paper really can be helpful to you all. So we're going to read the abstract, and then we're going to pull out some sections from the paper. All right? So let's read the abstract to start with. Since the first half of the 20th century, evolutionary theory has been dominated by the idea that mutations occur randomly with respect to their consequences. So what that means is that it doesn't matter, that mutations just occur wherever in the genome, and if a particular area shows less mutations than another area, then selection must be cleaning that area out when it gets mutations. Right? So that there's nothing in the genome that prevents mutations from occurring in certain areas. The only reason that certain areas don't have mutations is selection has been selecting them away. Okay? Here we test this assumption with large surveys of de novo mutations in the plant Arabidopsis thaliana. In contrast to expectations, we find that mutations occur less often in functionally constrained regions of the genome. Mutation frequency is reduced by half inside gene bodies and by 2/3 in essential genes. In other words, gene bodies, inside genes, mutation rate goes down by half. And in essential genes, genes that cannot be broken, mutation rate drops by 2/3. 66.6 or 67%. That's a lot. That's significant, right? With independent genomic mutation datasets, including from the largest Arabidopsis mutation accumulation experiment conducted to date, we demonstrated that epigenomic and physical features explain over 90% of the variance in the genome-wide pattern of mutation bias surrounding genes. Observed mutation frequencies around genes in turn accurately predict patterns of genetic polymorphisms in natural Arabidopsis accessions. R equals 0.96. That mutation bias is the primary force behind patterns of sequence evolution around genes in natural accessions is supported by analysis of allele frequencies. Finally, we find that genes subject to stronger purifying selection have a lower mutation rate. We conclude that epigenome-associated mutation bias reduces the occurrence of deleterious mutations in Arabidopsis, challenging the prevailing paradigm that mutation is a directionless force in evolution. Folks, did you hear all of what was said in there? 90% of mutation bias is explained by epigenetics and the physical features of how DNA is structured. 90%. That's nuts. That's crazy. I don't think you understand how big of a deal this is. There's a reason this paper got published in Nature. This is a big deal. All right, so let's pull some random sentences out of here and we'll talk about it. The random occurrence of mutations with respect to their consequences is an axiom upon which much of biology and evolutionary theory rests. In other words, this is foundational to evolutionary biology. And poof, up in smoke. This simple proposition has had profound effects on models of evolution developed since the modern synthesis, shaping how biologists have thought about and studied genetic diversity over the past century. From this view, for example, the common observation that genetic variants are found less often in functionally constrained regions of the genome is believed to be solely due to selection after random mutation, which is what I just explained to you a little while ago. Basically, their explanation previously for why mutations didn't occur in certain functional regions was because, well, selection was getting rid of them. Jumping down a few sentences here. Yet emerging discoveries in genome biology inspire a reconsideration of classical views. It is now known that nucleotide composition, epigenomic features, and bias in DNA repair can influence the likelihood that mutations occur at different places across the genome, and they cite 7 papers at the end of that sentence. All right, so they're not playing games here. They've done the homework, and I mean, this paper's not long. We're not short. We're going to read through a lot of it, but we're not going to read through a lot of it. But I want to talk about it because this is a big deal. Mutations not being random is a huge deal. Okay? It is a really, really big deal that mutations are not random and they're not constrained by selection. They're constrained by epigenomics and DNA structure. We'll talk about what that means a little bit more when we get to the end of the paper. Skipping down some more. Therefore, as expected, non-synonymous changes and premature stop codons accounted for a greater share of variance than in natural populations, and their frequencies were indistinguishable from a null model of random mutation. So in other words, they tested this against a random mutation model, and they had the same frequencies of approximately the same frequencies of non-synonymous mutations and random stop and premature stop codons that they would expect if mutations were random. Okay, so they're just testing it against that to see if their modeling and what they're doing is off in any way. Like that's all that was. So they are testing it against potential background problems. They are testing that, okay? So we're going to jump down a little bit more. We note in advance here that all downstream analysis led to the same conclusion for single nucleotide variants and insertions and deletions, or for germline and somatic mutations. So, in other words, in every case, single nucleotide variants, which is SNVs, indels, which is insertions and deletions, and both germline and somatic mutations, in other words, the mutations in the reproductive line and the mutations that take place outside the reproductive line, all were less frequent in gene bodies and essential genes. All of them. In other words, it doesn't matter where you go, if a mutation occurs, it is going to be more likely to miss the essential genes and gene bodies than it is to hit them. Every single time. Every single time. Okay? So we're going to jump down the paper a little bit more. Again, I'm just pulling out sentences here that are really interesting. Epigenome predicted mutation probabilities explained over 90% of the variance in the pattern of observed mutations around gene bodies. So in other words, the epigenome is allowing or preventing mutations in certain areas of the genome. That's really, really cool, folks. The fact that the epigenome is doing that, like, this honestly feels like something that Randy Galuza should write about, because this would fit really well in his model. I know some other creationists are a little more skeptical of epigenetics, and that includes me, but I think this might be a function of the epigenome that is worth talking about, because if the epigenome is preventing mutations in genes, and especially in essential genes, that's huge. That's a design feature, right? That points to a designer, somebody who sat up in heaven, in other words, the God of the Bible, and said, I am going to create this in such a way that the reprogramming outside the DNA that goes on, the epigenome, the above the or outside the genome, is going to control where the genome can be mutated. Folks, that's really, really cool. I know I'm a nerd, but that's really cool. Moving on. Since only 20 to 30% of gene body sites are estimated to be subject to selection, mutation bias in genic regions could affect sequence evolution around genes more than selection. Now, I highlighted this sentence for a reason, because the evolutionary world likes to talk about how the gene is the unit of selection. And I've argued for years that it's not. The selection can only see the phenotype. It cannot see individual genes. Yes, the individual genes are expressed in the phenotype, but unless they have large effect, they're invisible. Because, let's just say you have 3 different genes here, and you have a beneficial gene that increases eyesight, for example, and you have a deleterious gene that makes you slower, and you have a third gene that is just slightly deleterious. It increases your blood pressure just a tiny little bit. Now, if you are a lion hunting on the African plain, the fact that you have a gene that maybe cripples you or makes you really slow is going to be very visible to selection. It doesn't matter that your eyesight's much better, and it doesn't matter that your blood pressure's up a little bit. You just won't be able to catch food, so you'll starve and die. If you are if you're not a lion, maybe you're a wildebeest. Well, that extra eyesight might be great, but again, the fact that you're really slow makes you a target for a lion and an easy meal. And that cholesterol, selection can't see that at all. So, again, this is why I say that the phenotype is what gets selected for as a whole, not the gene. Because just that example right there should show you selection can't see all 3 of those. It can only see one. The one that has the largest effect, essentially. So, yeah, genes of large effect get selected for, and maybe they can be counterbalanced by other genes to a certain extent, but that all builds into a phenotype that has pluses and minuses in different areas and is then subject to selection on the whole thing, because it's not the gene selection is not just seeing the one gene with large effect, it's seeing the whole composition of the organism, and the gene with a large effect has to have an effect large enough to overrule all the other genes, essentially. So this sentence just I've highlighted because I think it debunks the whole Dawkins selfish gene idea. Not enough of the genome is subject to selection, only 20 to 30% of actual genes. This is not non-coding DNA. This is coding DNA. This is DNA that codes for protein, is directly associated with a phenotypic trait. And only 20 to 30% are estimated to be subject to selection. Which means that if their sequences change, if sequences of gene bodies change, mutation bias may be having a bigger effect than selection, according to this paper. Moving on. Genetic diversity in a global set of Arabidopsis accessions supported these results. Over 90% of the variance in polymorphism levels found around gene bodies could be explained by our experimentally observed mutation rates. In other words, the variance around the genes, the variance that existed in the organism, around the genes, not in the genes themselves, but around the genes, is explained. 90% of it is explainable by mutation bias. So in other words, the epigenetics at this particular site allows for more mutations, or the structure of DNA at this particular site allows for more mutations. Folks, that is huge. And they actually put this in their conclusion. In conclusion, evolution around genes in Arabidopsis appears to be explained by mutation bias to a greater extent than by selection. In other words, this mutation bias that they're talking about here is stronger than selection. It's having a bigger impact than natural selection is. And I think that's important because I think a lot of the times we overestimate the strength of selection to a certain extent. And we also, with this mutation bias, it's not quite directed mutation, but it's non-random. It's not directed. In other words, the cell is not telling the body's not telling the DNA, hey, make a mutation here or make a mutation here. But it is saying, hey, you can't make one over here. Go make one somewhere else. Essentially. Now, obviously, that's not quite accurate because there's no determinism to the mutations, right? There's nobody. But, the cell is essentially guarding certain, in particularly very essential genes, and preventing them from being mutated. Moving down just a little bit. Mutation probabilities were also 90% greater in genes lacking introns, and lower in genes with more introns and longer introns. Now that's really interesting because introns have function, right? We know they have some function, even though they don't get coded. If you don't know what an intron is, it's basically the section that is in a gene that is spliced out because it doesn't actually code for the protein. It's just it's a spacer, if you will. But apparently they have some function in reducing mutation rates in genes. Now I know there's some other functions for them, but that's the whole. They apparently help serve to reduce mutation rate. So if you have a gene that doesn't have a ton of introns, mutation rate is 90% higher. If you have a gene that has more introns, drops. Long introns, drops. Now that's interesting. I don't know why. They don't really go into why that is, but it's a very interesting observation and something that might be worth looking into later. Moving on. We next investigated mutation rates in relation to gene functions, discovering that genes with the lowest epigenome predicted mutation rates were enriched for conserved biological functions. For example, translation, in other words, translation of DNA into RNA. The gene that controls that, you're going to need that to work, right? You can't have that break. Highly conserved, not subject to high mutation rates. By contrast, genes with the highest predicted mutation rates had specialized functions, for example, environmental response. And this is all in figure 3a, which I will put on the screen for you all, because this figure is just amazing. This figure is just fantastic. We're gonna look at figure 3a. We'll just talk about it. I'll put it up on screen right now. But we're gonna talk about this figure because this figure is amazing, particularly the genes that are most subject and least subject to mutation. You see, there's two charts. There's a blue chart and a yellow chart. And these show the probability of mutations in a particular gene. The least likely to have a mutation, cytoplasmic translation. Then mRNA splicing, mRNA processing, RNA splicing, and two different types of RNA, actually all three RNA splicing genes are the next three. Very low probability of mutation there. Why? Because those are essential. You got to have those. You don't have those, things break. Which are the ones that are the highest mutational probability? Oh, response to chitin, cellular response to decreased oxygen or hypoxia. A lot of oxygen level response genes are very likely to mutate. Why? Well, because this is a plant. Plants use carbon dioxide. They don't really use a ton of oxygen. They make oxygen, though. So if their gene response to oxygen level breaks, meh, whatever, so long as it's not the carbon dioxide gene. But if their gene for translation in the cytoplasm breaks, that's a catastrophe. So it makes sense that the genes that are for things that are not quite as essential to life are the ones more vulnerable and not as protected by the cell. Folks, this is huge. This is huge. I keep saying that, but I want you to understand, this is a monster event. We now know that at least in this model organism, genes for particular important processes are more protected from mutation than less important processes. And it's not due to selection. They rule that out in the paper. I won't go into how they do that, but they rule that out. It's not due to selection. It's due to mutation bias from epigenetics and genetic structure, which means that the genome is built with foresight to prevent mutation to the most damaging areas. Wow. How great is our God to build something like that? That's incredible. This is the epitome of a design argument. This is incredible. Let's see. Comparing genes whose effects have been measured with knockout experiments confirmed that essential genes are enriched for epigenomic features associated with low mutation, and as predicted, observed mutation rates were significantly lower in the coding regions of essential genes. Folks, the genome was designed in a way that prevents mutations from doing as much damage as they could do. Even in a post-fall world, God is looking after his creation by designing them in such a way that they can react even when they're subjected to mutations. Here, moving on down a little bit. We found no evidence that the reduced mutation rate in essential genes could be explained by the potential intrinsic mutational properties of CG methylation, expression level, or GC content. Instead, the observed 37% reduction in mutation rates in essential genes is consistent with a reduction in mutation plausibly explained by their enrichment for low mutation-associated epigenomic features. Translation: the epigenome in the region of essential genes is structured in such a way that it prevents or reduces mutation. Folks, that's nuts. That is crazy stuff. This is I'm speechless. This is a design argument literally falling into our laps. Now, of course, they do talk about how it could have possibly evolved. Our findings reveal adaptive mutation bias that is mediated by a link between mutation rate and epigenome. This is mechanistically plausible in light of evidence that DNA repair factors can be recruited by specific features of the epigenome. Hypomutation targeted to features enriched in functionally constrained loci throughout the genome would reduce the relative frequency of deleterious mutations. The adaptive value of this bias can be conceptualized by the analogy of the loaded dice with reduced probability of rolling low numbers, that is, deleterious mutations, and thus a greater probability of rolling higher numbers, that is, beneficial mutations. So they're arguing here that this will allow for more beneficial mutations because it will decrease the frequency of deleterious ones. Okay, theoretically that's possible, I presume. But you're making the assumption here that the areas where the mutations occur don't have some kind of function, and that they aren't nearly neutral mutations that Kimura talked about, where they are slightly deleterious, very small amount of deleterious. That's assumed here. And further, this is not a mechanistic explanation. This is, well, theory should work this, it should work this way, but there's no mechanism here. There's no mechanism for how this could have evolved. None. It's not there. So, and there's a theoretical model that they go into. Again, there's no mechanistic explanation. There's some theoretical modeling, and that's fine. That's interesting. But there's no mechanistic explanation. Not that I'm surprised that there never is, or very rarely. But in situations like this, it's just shocking to me that they have undercut a very large basis of evolutionary theory for the past, I don't know, since the modern synthesis in like the 30s and 40s, like maybe even earlier than that. I don't remember exactly when the modern synthesis was formed. But like this has been, they've just bashed that right out of existence. And they're still trying to fit it into evolution somehow. How about we look at this as a design protection, where God knew that creation would fall, he would place the curse, mutations would start to happen, but he wanted his creation to succeed and thrive, and so what does he do? He builds into creation a beautiful, amazing guard for mutation, to prevent mutations in the most essential areas. Wow. You think about it that way, it's paradigm shifting. So that's all I've got. 25 minutes on this paper. It was a fun paper to read. I will link to it in the video description. It is open access. So all of you who are amateur genetic nerds can feel free to go read it. I will link to it in the video description. I think it'll be a great fun. Please, if you like the content that you're seeing, subscribe. Follow us on alternative tech platforms. The things we say don't tend to be popular, and unpopular things are getting banned. So, follow us on Gab, follow us on Odyssey, which is where we're putting a lot of video content, Rumble, etc. And we do occasional one-offs on those other platforms, which are about things that would get us banned from YouTube. So feel free to check us out there. Also check out our website, inhisimage.blog. We do have a Facebook page and a MeWe page as well, and a Parler and a bunch of other social medias. So that's all. All the social medias are in the video description. Feel free to check those out, and I look forward to seeing you all again next time.